Today, we'll be discussing range of thallus organization in algae. Range of algal thallus varies from unicellular to multicellular forms or microscopic to macroscopic structures, with their size ranging from a few microns to some meters. Micromonas pusilla is known to be the smallest algae, which is unicellular and is 1 micron meter. On the other hand, giant kelps has longest uh, thalli that reaches up to 60 meter in length. The unicellular forms may remain solitary as a single unit, which are capable of completing their life cycle by providing all physiological, biochemical, and genetical requirements, and may be motile or non-motile. When these unicellular forms are held together in a common gelatinous matrix, they constitute colonial form, which are considered intermediate between unicellular and multicellular structures. First is unicellular forms of algae. Unicellular or acellular forms can be motile or non-motile and are further grouped into three categories based on the presence or absence of flagella. The motile forms are either rhizopodial or flagellates, and non-motile forms are coccoid. Rhizopodial forms lack rigid cell wall and have a naked protoplast. Cell envelope is periplasmic, soft, and permits extensive changes in shape and size of thallus. It lack flagella and instead possesses cytoplasmic projections called pseudopodia and rhizopodia. So these forms move in amoeboid manner. So just look at this first diagram. This is an example of rhizopodial form, and this example is Chrysamoeba. Second is flagellates. The vegetative phase of many alga is a motile flagellate unicell. Flagella may be one or two or many, equal or unequal, and tinsel or whiplash type. Motile unicells are commonly spherical, elongate, ovoid, or round in cross section. Flagellated forms are present in almost all groups of algae except Myxophyceae, Phaeophyceae, and Rhodophyceae. So you can see here example of flagellate algae, Euglena. Next is coccoid form. Non-motility here predominates and motility is entirely absent or restricted only to reproductive phase. It occurs in majority of algal classes and predominates in Xanthophyceae. Means 70% of members of Xanthophyceae are coccoid form. Coccoid forms are provided with rigid cell wall and are non-flagellated. Example is Prochloron. So this is an example of coccoid form. Last form is spiral. Spiral is unicellular and having spiral filamentous body. Example is Spirulina. Second form of algae are multicellular forms. Multicellular forms again are of four types: colonial aggregation, then filamentous form, siphonocladus organization, or siphonous organization. So first we'll start with the colonial aggregation. A colony, is a group of separate cells, generally similar in structure and function, and aggregated by a miscellaneous envelope. The colonial habit can be further differentiated into synobium, palmilloid form, tetrasporal, dendroid, or rhizopodial. Synobium has a definite number of cells arranged in a particular manner, which is determined at a juvenile stage and does not increase during its subsequent growth, even though the cell enlarge. It comprises aggregation of flagellate cells or non-flagellate cells. Just look at this first diagram. It's example of synobium with flagellated cells. Example is volvox. And non-flagellate cells example is hydrodiction. Second type is palmilloid form. Colonial members in palmilloid form have non-motile cells. When non-motile cells remain embedded, here not the shape and size of the cell is constant. The cells are aggregated in a common miscellaneous envelope. All the cells are independent of each other and fulfill the function of an individual. So most Commonly means normally flagellate or coccoid unicellular algae may enter uh, Pamela stage, means a condition where the flagella are lost and the individual undergo successive vegetative divisions while embedded in a common gelatinous matrix. So this is named after the volvocaline genus Pamela. So this term may be strictly applied to those algae where the cells will radially revert to a motile condition, or may be expanded to include all algae where the pameloid habit is more permanent. So example is Pamela. Just look at this second form. This is an example of pameloid form. Next is tetrasporal. Here, motile stages are restricted to reproductive cells only. The regular colonies of Synechococcus, uh, example example is Merismopedia, as flat plates of cells. Just look at this third diagram. And Eucapsis with colonies in cubical masses, so are included in tetrasporal type. So a feature of tetrasporal form is the presence of mucilaginous pseudocilia. So this is an example of tetrasporal form. Next is dendroid. Just look at this next diagram. Dendroid. Here cells are united in a branching manner by localized production of mucilage at the base of each cell. Then the whole colony looks like a tree in habit. So example is Chrysodendron. This is an example of dendroid form. Last is Rhizopodial form. Example is Chrysophyceae. Here the cells of rhizopodial colonies are united through rhizopodia. And you can see here, these are rhizopodia, and here the rhizopodial colonies are united through these rhizopodia. Next form is filamentous form. Filaments are formed when vegetative division occurs in a transverse plane. In a filament, the uniciliate row of cells are joined end to end in a transverse plane through middle lamella. In Cyanophyta, a filament includes both the trichome, that is the uniciliate row of cells, and its sheath. The filament may be branched or unbranched. Unbranched filament, for example, Ulothrix, Spirogyra, and branched filaments, example, Cladophora or Pithophora. So, in the case of branched filaments, branching may be false or true. False branching occurs in some members of Cyanophyta. Here, the trichome generally fragment due to the degeneration of an intercalary cells or by the formation of biconcave separation discs, after which one or both of its end adjacent to the dead cell grows out of the parent sheath, giving the resemblance of branching, right? In the case of true branching, it results from repeated transverse division of the lateral outgrowth produced by a few or many scattered cells of the main filament. The truly branched thalli are of four types. First is simple branched filament. Second is heterotrichous. Third is parenchymatous form. And last is pseudoparenchymatous form. Simple branched filament, here a branched upright filamentous thallus is attached by a simple disc derived from basal cell. For example, in the case of Cladophora, just look at this diagram. Then heterotrichous form. Here the thallus consists of two parts. Example is Ectocarpus. Just look at this second form. Heterotrichous. Example is Ectocarpus. So here thallus consists of two parts. A prostrate creeping form, creeping base, and an erect branched upright system. It is the most highly evolved type of filament construction. During its development, a system of branched creeping filaments, it produced first Then, and it functions as a holdfast. These creeping systems of filaments will further give rise to a system of erect and branched filaments. Right? So, example here, ectocarpus. Next, third type is parenchymatous form. This occurs when cells of the primary filament divides in all direction. Any essentially filamentous structure is this then lost early. For example, alva. Next, its last is pseudoparenchymatous form. This habit results from a close juxtaposition of the branched filaments of a single or many axial filaments. If branches form a single filament are involved, as in the case of Batrachospermum, just look at this last diagram, Batrachospermum. When branched form a single filament, as in Batrachospermum, the thallus construction is called uniaxial. If branches of many axial filaments aggregate, and thallus is multiaxial. Example is Nemmelian. Next form is Siphonocladus organization. It is restricted to members of Chlorophyceae, in which unbranched, for example, this Hetomorpha. It is restricted to members of Chlorophyceae, in which the unbranched form, for example, Hetomorpha, or branched, for example, Cladophora. Filaments are composed of multinucleate or semicenocytic cells. One of the Siphonocladus alga, for example, Valonia, occur as a single spherical vesicle up to 10 centimeter in diameter and has been described as the largest plant cell. Right. Last is siphonous organization. Here, in a number of marine Chlorophyceae, members of Chlorophyceae, for example, order Siphonales, etc., and some Xanthophyceae, example, Wucheria. Here, enlargement and elaboration of thallus proceeds in the absence of septa. Just look at this diagram. This is the structure of Wucheria. Here, no septa. You can see here, no septas are present. So, nuclear divisions are not followed by cytokinesis, means free nuclear division, and the result of a senocytic multinucleate thallus and a siphonous organization. Right? Example is Wucheria. Thank you.